This is my logo, but what it is, and what are these? For those who are curious, these are the differentiation stages that happen to the malaria parasite after it invades the mosquito midgut. And now I'm going to show you how I came up with this from this. So I was making a logo for this channel and thought that it might be interesting to show you how I was doing it, mainly because I'm making it in a sort of different way as how logos are usually made. I have to mention that I'm not a graphic designer, and I am quite bad at Java. But what I am mildly adept in is image processing and analysis. Therefore, I thought that this video might come in handy to make a brief introduction to some basic image analysis tools and concepts. These are my starting images. I took them in an experiment where I was determining the relative concentration of a certain molecule during the parasite differentiation in vitro. The first one is the bright field image. The second one is a fluorescent image using a fluorescent probe for glutathione, a fairly abundant molecule in the cytoplasm. The third one is also a fluorescent image, but for the nucleus of the parasite stained with DNA-specific fluorescent dyes. The other file contains some regions of interest or selections of the parasite that I did previously and which we might use in a moment. As you see, what I want to do for my logo and banner is to make simplified images of the differentiation stages, highlighting some characteristics of the parasite. I want them to look membranous and to clearly show the nucleus, which makes them look actually like cells and not just nodules. Then I want to represent the malaria pigment, these black spots, which are a specific characteristic of these kinds of parasites. To achieve all this, I'm going to use ImageJ. ImageJ is a free and open source software for image analysis widely used in the biological sciences. It's tiny, just this little window, but it's super versatile and powerful because you can expand it as much as you need with macros and plugins, making it really fun to use. Okay, so the first thing we are going to do is to identify the parasite, or rather we should say, delimit and isolate it from the rest of the image. This process is called image segmentation, which is nothing more than to partition an image into regions according to what interests us. In this case, our region of interest is the parasite. But we don't just want the parasite outline. That would be boring. We also want to preserve some features inside the parasite to make it more interesting. We could use the bright field image, but we don't want the malaria pigment showing right now. That will be for later. So we are going to use the fluorescence image. Now, there are many segmentation methods that exploit different features of an image, and their implementation depends on what we are interested to highlight. For this case, I'm going to use an edge detection filter, specifically a Laplacian of Gaussian or log filter, also known as a Mexican hat filter because it resembles that, a Mexican hat. This is the radius of the filter we're going to apply. The bigger the radius, the smoother the resulting images will be. Now we have a much simpler image of the parasite and some internal features clearly defined. The Laplacian of Gaussian operator gives us the opportunity to introduce yet another important concept in image analysis: convolution. Convolution is a mathematical operation on its own that combines two functions to produce a third one. It is like an amalgam between addition and multiplication that modifies our function, in this case an image, with another function or kernel, in this case our log filter. So the output is a new image that has been reshaped by the filter. Okay, so a digital image is a matrix of pixels, okay? And the filter is also a matrix. So basically, convolution is applying one matrix to another one. Let's exemplify how it's done. This is a Laplacian kernel, and suppose this is a random image. We want to filter this image with this kernel, so we convolve it. Convolution is performed stepwise by sliding the kernel over each pixel of the image, defining its new value. First, we superimpose the kernel over the first pixel and multiply each coefficient of the filter with its corresponding pixel in the image. However, there are no values outside of the image. How are we going to perform any operation with absent values? The way to deal with this is to add extra pixels in the border. We could just treat the absent values as zeros, which will be black, or extend the border pixels outwards in each direction, or we can mirror the image to the borders, or wrap it all around in each direction also. There are multiple ways to deal with this, and how far the borders are extended depends on the kernel size. It is suggested that the borders need to be extended at least half the size of the convolution kernel. For now, we are just going to extend the outer pixels, and one row is enough because our kernel is 3 by 3. Okay, but returning to convolution, we multiply each pixel with its corresponding pixel in the kernel. So this one times this one, and this one times this one, so on and so forth. Then we add all the products, and that's the new value for our pixel. We then slide the kernel one pixel, repeat the process, and continue like so for the whole image. That's convolution in the context of image analysis. For me, it's the single most important operation one needs to learn, as it is useful for removing noise, blur or unblur an image, detect edges, and many more and way impressive things like super resolution microscopy. Interestingly, turns out that the log filter is actually the convolution of two different filters. A Laplacian filter, which is the one that actually detects the edges, and a Gaussian filter that smooths the image. Smoothing the image is an important step because edge detection filters are very sensitive to noise. For example, here is the parasite image convolved with only the Laplacian filter. So the image is smoothed first and then the edges are detected, okay? However, convolution is commutative and associative, meaning that the order in which the filters are applied and furthermore, which filter is applied first or last and relative to which one does not matter. In fact, instead of convolving the image with a Gaussian kernel to smooth it, and then convolving the resulting image with the Laplacian kernel to detect the edges, or vice versa, we can convolve the Laplacian kernel with the Gaussian kernel to obtain the Laplacian of Gaussian kernel, which is then applied in just one step to the image. But let's stop babbling and demonstrate. Here I'm representing these kernels in image form. And if we convolve the Laplacian with the Gaussian, we get the log filter. Now let's apply these filters separately to an image. For testing, I like to use standardized images like Lena. However, she already retired, so we are going to use Rumble instead. We are going to apply the log filter first, and then we are going to apply a Gaussian filter, followed by a Laplacian filter, and finally, we are going to apply the Laplacian filter, followed by the Gaussian filter. All three resulting images should end up being the same. Doing these convolutions manually in ImageJ is not that straightforward. There are some small details that need to be addressed. And here I would like to thank Dimitar Trodanov and especially Herbie for pointing me in the right direction. Okay, because there are a lot of steps, I wrote a little macro that will perform all of them really fast. So let's run it. First, we add a zero-evaluate border, half the size of our biggest kernel, to prevent border effects during convolutions, okay? Then, we separate the image into the three color components, red, green, and blue, and convert each one into 32-bit images. This step is needed because the log filtering produces pixels with negative values. Actually, the Laplacian filter has a -4. That's the source of the negative values. But wait, that's not possible. Images normally have pixel values that range between 0 and 255. Well, this is because we usually work with 8-bit depth images, which is more than enough to represent all visible colors. But 32-bit floating point images can actually have negative valued pixels and a really much pixel depth. Okay, so once the colors have been separated and the image is converted to 32-bit, we then apply the log filter to each color channel, convert them back to 8-bit images, and merge the three channels back together. And here is our log filtered Rumble. Now, let's let the macro perform the convolutions for the separate filters and in different order. First the Laplacian, then the Gaussian, and then the Gaussian followed by the Laplacian. As you can see, all images are indeed the same, or pretty much the same, since there are some errors introduced by rounding and converting back to 8-bit. Look at those cars. Okay, that was a lot of information, right? But hang on, we still need to process the two other images. Oh, but first we have to convert the parasite image into grayscale. We don't need the color anymore. Now we are going to do the malaria pigment segmentation from the bright field image. This image is quite complex. There's a lot happening here. However, since we are only interested in features that are inside the cell, we can add the ROI of the parasite to this image and clear the outside. We can see that it is now a much simpler image. So then we convert it to 8-bit because right now it's in RGB format, despite being grayscale. And for segmenting the pigment, we are going to use a different approach. We are going to use a method called thresholding. This type of segmentation replaces each pixel in an image to black or white depending on a certain intensity threshold we assign. So all pixels below that threshold will be set to black, and all pixels above that threshold will be set to white. So if our image has a sharp contrast between what we are interested in and the background, our region of interest will be segmented. Thresholds can be set manually, but there are actually a lot of automatic thresholding algorithms. Most are based on properties of the image histogram, but others use different approaches like calculating the mean intensity and standard deviation, and then defining mathematically a threshold from there, or by iteratively clustering an image into two classes, foreground and background, until certain parameter in the resulting image remains constant, for example. We are not going to delve into this because I'm going to use the one that works better. And for our needs right now, that means the one that makes the prettiest dots. The moments thresholding seems okay, but I'm noticing that the edges of the malaria pigment are a bit noisy or irregular. So for that, I'm going to apply a median filter with a radius of 5. The median filter is just that, the median of the pixels within a certain radius, which results in a smoother image with reduced noise. So if we apply a median filter with a radius of 5 to our hypothetical image, we can see that the image is less noisy and the border is better defined. Returning to our parasite, there's another problem here. The malaria pigment is being considered as background, while the parasite is the foreground. To solve this, we are going to select the magic wand and add all these pigment spots to the ROI manager. The magic wand is based on a concept known as region growing. Basically, a seed pixel or a small region is selected, and then this seed starts growing to adjacent pixels if and only if these pixels meet certain predefined intensity threshold. So if a neighbor pixel has an intensity value that exceeds the threshold, the region will not grow to that pixel, allowing us to select only the pigment spots in this case. So I'm going to add all these spots to the ROI manager and combine them into one. Then we can delete the single ones. Now, with the pigment spots ROI selected, let's clear the outside, fill the inside, and invert the image. And there you have it, the pigment spots have been segmented. Last, we need to segment the nucleus. I'm going to segment it by also thresholding the image. And for that, let's turn the image to 8-bit and find a threshold algorithm that works fine. Moments, the default one. Intermodes. Yeah, intermodes works fine. It's a bit smaller. Also, this image does not seem to have a bimodal histogram. The intermodes algorithm finds a threshold value by averaging the two modes of a bimodal histogram, but whatever, it looks fine. Again, I'm going to remove a little bit of noise and invert the image. Now I'm going to save all these images and open the next ones, which are from here to here. And you get the idea. I'm going to proceed with the rest of the images. All right, here are processed images, and now we are going to move to another program, which is Inscape. Okay, here we are going to import the images and apply some further processing. Right now, the parasite is a solid blob, but I want just the outlines of all these structures. So we are going to trace the bitmap with an edge detection algorithm that will convert this image into lines. No, edge detection. Here, the threshold only means the thickness of the lines we obtain. 0.5 seems fine. Here, Inscape is applying a multi-step algorithm called the canny edge detection, which is fairly complex, and since this video is already a bit long, we will leave the explanation for another time. Also, I could have obtained the outlines in ImageJ, but I did it in Inscape because here I'm actually vectorizing the image. That is, converting it from a raster graphic to a vector graphic. While a raster graphic is comprised of pixels, a vector graphic is rather comprised of vectors which trace a path, a series of points or vectors connected by lines or curves. This means that the image can be zoomed indefinitely without ever becoming blurry or pixelated. This is the choice of graphics to make logos, for example, or for computer-aided design and 3D printable models, for example. Okay, so now the nucleus. For this one, we also want to detect the edges. And finally, the malaria pigment. But here we want to use a brightness cutoff because we are not interested in the edges. Again, this looks the same, but now this image is actually made out of vectors. It's no longer a bitmap. Now we are going to move a little bit faster because all the proper image analysis has been left behind. Right now I'm just aligning everything and proceed with the rest of the parasites. So here we have all our parasites, and what I'm noticing right now is that the images are still a little bit too complex. For example, this is the first parasite I processed, and it became much simpler and nicer, which is probably because it came from another batch of images taken in another experiment. But what I'm going to do is just to remove some vectors of the image to look them less cluttered and to delete some of the surrounding stuff. While I'm doing this, notice all the vectors that make up the image. All those dots are the vectors that are connected by lines. It's like those puzzles where you have to connect the dots to reveal a shape. I will leave some surrounding stuff because I think they look cool. So now I'm adjusting some of the shapes to make them look nice. You know, we need to favor the looks. It does not need to be scientifically accurate. I mean, this is for my logo and for my banner. Now I'm moving the parasites to their proper developmental order, and I have already chosen my color palette, so let's color them. At the end, I decided to add 2 additional differentiation stages. One previous of the banana-shaped one, and another one after the spherical one, just to fill the available space for the banner. And here we have it. I hope you find this interesting and gave you a glimpse into what image analysis is. Thank you for watching.